Sir, could you please start recording?
Yeah, I did it. Why I have so genetics is in the lecture you saw. So genetics is basically all study of genes, which controls the heredity. And in order to understand genetics or to study heredity, it's very important that we do have variation in genes. Unless we don't have variation in genes, we won't be able to do some genetic analysis. So Gregor Mendel, so today's lecture is about, so this lecture which I uploaded 2 days ago was about Mendelian genetics, we call. And Mendelian genetics is classical genetics which led to discovery of factors which control traits. The question in front of Mendel was that in 17th century, the Spanish wool was of much higher quality and they were conquering all the market. And people from the intellectuals or scientific community gathered together in Austro-Hungarian Empire, and they decided to think about how to improve the quality of their wool. So that was the fundamental question behind what Mendel achieved. Now, in order to understand what controls the quality of wool, you can imagine 200-300 years ago, nobody had an idea about genes or any factor that may control a specific trait like quality of wool. So Mendel decided to use pea plants, and he could see that he already has variation in pea plants. So he had round seeded plants. He had wrinkled seeded plants. He had purple flowers, white flowers. He had yellow color of seeds versus green color of seeds. So he had these traits which were very clearly different in different plants. And he thought, if I can understand what controls the round color or green color of seeds or yellow color of seeds, I may come up with the idea about, how to improve the wool quality. But in order to do such a big experiment to improve quality, you have to first understand and find factors which actually control different traits an organism has. Now, Mendel proved to be an extremely smart fellow. What he did, he said, before I chase these factors, let me first see if I grow round seeded plant, if generation after each arrow is a generation. If generation after generation, I have round seeds and no mixing of any wrinkled seeds. And same is for if I have wrinkled seeded plant and if I grow them generation after generation, there's no contamination of round and it always in 100% cases produces wrinkled seeded plants. Now we call such parents or such progeny or population, we call them pure breeding lines, pure breeding, or true to type. Round is always going to produce round. Wrinkled is always going to produce wrinkled. So such experiments in biology, we call such experiments control. Control, it serves as a reference for our experiment, because what we see is that we have a material which is always going to produce either round or wrinkled. If the parents were round, it's always going to produce 100% rounds. Then he started doing experiments and he said, okay, so I'm going to cross a round pure breeding or true breeding round with a wrinkled pure breeding parent. What he discovered that in the progeny, all he found was everything round. Wrinkled disappeared. So this trait, it disappeared and round dominated. When he grew now, and this generation is called F1. When he crossed this F1, this sign means self-crossing. When he took pollen grains of round and threw on the stigma of the same plant, what he discovered that there were rounds and wrinkle came back without any loss. And in the F2, so this is now the F2 population progeny, the ratio between round and wrinkled in F2 turned out to be 3 rounds and 1 wrinkled. He did these experiments with other traits, like let's say he took yellow seeded, crossed with green colored seeds. He went to F1, and in F1, what he discovered that everything was yellow. He self-crossed this, and what he discovered that in F2, he had 3 yellows and 1 green. He did these experiments with 7 different traits. I told you some of the traits I explained you here, you know, the seed color, the seed shape, flower color, then the height. He also took, let's say, tall pea plants, crossed them with dwarfs, and in F1, what he discovered, everything was tall. He self-crossed F1 tall, and what he discovered that again, the trait which disappeared in F1, which was dwarf, it reappeared, and the ratio was 3:1. So the trait wrinkled seeds or green colored seeds or dwarfs, the one which disappear in F1 and the one which dominates because it was 100% rounds. So Mendel coined 2 terms. So round was said to be a dominant trait, and the one which disappears and reappears without any loss in F2 was called a recessive trait. Okay. Is it clear so far? So we have a dominant trait which masks the wrinkle, and the one which disappears in the F1 is called recessive. Now what he did, he,
Excuse me, sir.
yeah.
So in the lecture you said कि जो round वाला था वो भी true breed था, क्योंकि जब उसने round को सारे को किया था तब round आ रहे थे.
Yeah.
और जो wrinkle था वो भी true breed था, क्योंकि जब उसने wrinkle को किया था तब भी सारे wrinkle आ रहे थे.
Yeah.
तो अगर दोनों true breed है तो हमें कैसे पता चलेगा कि dominant कौन सा है recessive?
Because we crossed, so this is, let's say, father and this is mother, And in the progeny, we took seeds of the cross, we grew them. Now these seeds, all these seeds are from progeny of this cross. If they all are round, that shows wrinkle disappeared in F1. Clear? Is it clear? Who asked the question? Hello?
Sir, I did. Yes, it's clear.
Is
It's
it
just
clear?
हमें तभी पता चल yes sir. So हमें तभी पता चलेगा ना जब हम उनका आपस में क्रॉस करवाएं and we see which phenotype is appearing.
Yeah, absolutely, absolutely. Ji, who else has question? Amna?
Sir, I have a question.
Ji, Ibrahim. Who?
Okay, sir.
Go
Ibrahim,
on.
yes, Ibrahim. Okay. So, sir, ये हमने we did some experiment with uncrossing and we get some results, and now we are analyzing them to know what kinds of gene are recessive and what kind of gene are dominant.
Yeah.
But for example, some traits are they may take a while to observe. Like for example, if we are measuring a height of a tree from when it was a seed, it may take up to 100 years. So with subjects like physics, we can simulate models like car crashes and stuff like that. So is there a way in biology where we can simulate such genetic experiment to speed up the process and maybe make it more accurate, reliable, and more flexible?
Very good question. So the field of modeling and simulations is just entering into biology. It's very fresh, but I think let me tell you, biological system is surprisingly highly unpredictable. It unfortunately so far, I mean, we can predict, but we cannot be sure that the biological system will work that way. Gee, who else has a question? There were 3 hands raised. Okay, so what it did in F2, and this is something which was really interesting. He randomly took 100. So in F2, you have rounds and wrinkle. Let's say we have 100 total F2 individuals, and roughly you will have 75 will be rounds and 25 wrinkle. That's what makes 3:1 ratio. Now, what he did, he randomly chose round seeds, wrinkled, round, wrinkled, round. He randomly grew from F2 and then went to see what kind of progeny they produce. Because here, what is clear, there is very distinct 3:1 ratio. And round is, in terms of genetics, what precise word will you use, the term you will use for round or wrinkled? This is appearance. This is physical appearance. So what terms will you use?
Phenotype.
So phenotype. This is very important point. So 3:1 is a phenotypic ratio of F2. F2 phenotypic ratio is 3:1 in a monohybrid cross. We are doing a mono. Mono means single, and hybrid when you are using 2 different parents. When you use 2 different parents, you are chasing only one trait, you have 3:1 phenotypic ratio. However, when you used F2 random 100 individuals and you try to see what kind of progeny they have, what he discovered, the ratio turned out to be 1:2:1. 1 pure round, 1 pure wrinkled, and 2 impure rounds. I repeat, you took, let's say, 100 F2, you grew them individually, and you looked at their progeny, what he found that, okay, this round will always produce round. No mix of wrinkle. Everything is round. This one produces 100% wrinkle. No mix of rounds. Okay. This round produces 3:1, 3 rounds, 1 wrinkle. This wrinkle always produce wrinkle. No rounds. But this round may produce, and when he counted all of them, he said, okay, there is a ratio 1 pure round, 2 impure rounds, and 1 pure wrinkle. This makes, it means the 3 rounds, phenotypic rounds, which we see here, one of them is pure round and 2 will be impure rounds. G, there are 2 hands raised. G.
सर ये क्वेश्चन है कि ये जो 3:1 दिस वाज phenotypical ratio, so ये जो 1:1 है, ये तो उनकी जीन्स में रेशियो है ना, क्योंकि ये तो नहीं शो होगा phenotype में.
Yeah, so we have 3. So let's go slow a bit because not everybody in this class knows how to link this phenotype with the genotype. And that's why intentionally I haven't used the word gene or genotype. Okay. Now, आपका नाम क्या था जिसने सवाल पूछा था?
Abdullah.
जी, Abdullah. Abdullah, so next few steps will tell you how we link this 1:2:1 with the phenotype. Because so far, we are just trying to play Mendel by ourselves, each one of us. And we are trying to see how we go to the genes, because so 200-300 years ago, nobody knew about genes. So how Mendel solved this question? He saw all of this highly reproducible, same 3:1 phenotypic ratio if you use yellow versus green seed color, tall, dwarf, or purple versus white flower colors. 3:1 is the F2 phenotypic ratio, and 1:2:1 was the ratio 1 pure green, 1 pure green, 1 pure yellow, and 2 impure yellow. Okay, because yellow he discovered was dominant, green was recessive, tall was dominant, dwarf was recessive. So once let's write everything now more neatly here. Round versus wrinkled. F1 was all round. Self-crossed F1, we had rounds versus wrinkled. What we said, round is dominant, wrinkled is recessive, and in F2, after self-crossing F1, we found 3:1 ratio of round versus wrinkled in F2. When he grew F2 individually, what he found, 1:2:1, so pure round, pure wrinkled, and impure rounds. We said 3:1 is phenotypic ratio of a monohybrid cross. Today we are going to do monohybrid cross.
Okay. Sir, I have a question.
G, please.
Sir, what would be the genotype of the improper round?
What was that?
What would be the genotype of the improper round?
I am Mendel. I don't know at this moment. I'm going to decipher now. Isn't it what we are learning now?
Okay, sir.
So, to all the students who have never taken biology before, or even the ones who have taken but have forgotten the fundamental concept, now what Mendel did, he had these phenotypes and he had these results. He studied 7 different traits, we said, 7 different traits. And in all those traits, he found same ratios into play. These were not precisely 3:1. They were like 2.97, 2.8 is to something. And he rounded everything and he said it's most likely 3:1. Now, remember what was his question? He wanted to understand the factors which control these traits, the phenotypes, the wool color. He just forgot wool color for a moment and he said, let's first think about how traits are controlled in a living organism. He postulated, he said, you know, this round trait is controlled by a pair of factors. I repeat, he postulated that the round trait is controlled by a pair of factors and he started giving them symbol. He said the one which is dominant, round, I'm going to give capital letter R. And the one which is recessive, I'm going to represent it with small letter r. And his real hypothesis was these factors, they exist in pairs. That's why he wrote a pair, capital R, small r, small r. The next thing was very important. He said in an individual, the factor exists in pairs, but when a parent is going to pass factor to its progeny, its babies, it can only contribute half of the factor, which means just a capital R. And this one will only contribute a small r. What will be in the F1 now? This round will be capital R, small r. This individual. And he's already coined the term dominant. He said, since round is dominant over wrinkle, the factor which gives us round, capital R, is masking or dominating the small r. That's why we have round phenotype in all F1. Now, he said, okay, I now self-crossed, which means same plant, pollen grain of same on the stigma of same. So what I have here now, although the phenotype is round, but the factors are different in this round, F1 round. Now, what we said, when they will go to next progeny, only half of the factor can be contributed. And we draw a checkerboard or Punnett square, we can say. Now, there's no restriction. Either one of the two factors, either this one or this one can go. So he separated them. And there is no restriction on which one will mate with what. So if capital R combines with capital R, this is also possible, this is also possible, and this is also possible. What is the phenotype of this one? Tell me phenotype of this one, which is being encircled.
Round.
What is phenotype of this one?
Round.
Round. And this one?
Wrinkled.
Wrinkle. So what do you have now? 1 round, 2 rounds, and 3 rounds, and 1 wrinkle. What did we get in F1?
3:1.
3:1. He had, I mean, these are not the small numbers. He got numbers in hundreds, 800 something rounds, and 400 something or 300 something wrinkled, and the ratio was turning out to be 3:1 or 2.98 to close to 1. Now, these rounds are represented by capital R, capital R, capital R, small r, and wrinkled is this one. But since we are looking at phenotype, he was getting just the phenotype and he got 3:1. When he started postulating all this, that's why I call him a man ahead of his times, a super genius. He had no idea about chromosomes existing in pairs, genes existing in pairs, homolog chromosomes. He had no idea, but he postulated all these things. Now, when he took these F1, F2 randomly, you pick all of them randomly, what he discovered that I get one pure round. Which one of all of them will be pure round?
Capital R, capital R.
Capital R.
Very good. So this is the pure round. These two are definitely, when you will grow them, this is like this progeny. So if you take F2, these rounds, it is exactly like F1 progeny. This turns out to be an impure round. This is also impure round. So these are two impure rounds, and this one is pure wrinkled. Is it clear so far?
Yes, sir.
Okay, now somebody earlier used the term genotype. And we also use this term in our earlier lectures. So what Mendel discovered was, he said, traits, they are controlled by factors. Factors exist in pairs. And when a parent contributes to the progeny, only half of the factor goes to the progeny. Okay. And one of the factor is dominant, will be dominant over the other, which he said is recessive. What he discovered genotype for phenotype. Now, when we have capital R, or small r, we call them homozygous. Homozygous round, homozygous wrinkled. Remember this term. Homozygous recessive. If I say homozygous recessive, you should know immediately Tariq is talking about wrinkled. Small r. Homozygous round, you should immediately know Tariq is talking about capital R. Homozygous tall, homozygous dwarf is recessive. So you should know all this. Now, what is this individual then called? This is called heterozygous. So what Mendel proposed was that factors, they segregate randomly, they segregate independently. And so like here, factor exist in pair, but each of these two, capital R, it means this is what I should have written. Just like here, this parent, a heterozygous round, it can contribute either capital R or it can contribute small r. That's how such a progeny 3:1 will be possible. And similarly, if we have homozygous recessive, small r, this parent can contribute, both the factors will have equal probability to go and contribute to the progeny. But of course, since it is homozygous, we have small r. Any questions so far? J. Ibrahim.
Okay, sir. So I have a question related to the pairing of alleles. For example, if two different kinds of alleles exist, it means they may have been mutated in a stage in life. That's why the two different copies of genes exist, alleles exist. So for example, let me talk about an example. Let's say my father's blood group is OO and my mother's blood group is AB. So how is it possible that I have a O blood group, which is a recessive blood group? Like it is possible that the A allele or the B allele got mutated.
So we'll talk about, so you're talking about when you bring in blood groups that, so your question is very good. The first part of your question. When you bring in the blood groups, you are bringing in example, which we will discuss maybe 2 lectures down, and people will get confused. And we call that phenomenon codominance. Okay. Today we are talking about only and only complete dominance. Complete dominance. Complete dominance is what? Whenever round and wrinkled are together, tall and dwarf are together, yellow and green are together, one of the factor is completely dominant. Round is completely dominant, tall is completely dominant, and yellow is completely dominant, or purple is completely dominant. Blood group is an example of codominance, where you have two alleles coming together. And why I like your question, because you are bringing me exactly to the point where I wanted to come on next slide. What is capital R and small r? Yeah, please ask question. Who want to ask question? There are 2 hands raised.
Sir, I want to ask question. Sir, I want to confirm कि जब हमारा जो रिसेसिव एलील है वो तभी ट्रेट शो करेगा जब वो होमोजाइगस होगा. तो जो
Yes.
हमारी F2 के अंदर हमने जब ब्रीडिंग करवाई है, we got 1:2:1, तो we will either have heterozygous or homozygous, right? तो heterozygous के अंदर जब हमारे पास डोमिनेंट एलील आ जाएगा, तो हमारे पास वो तो रिसेसिव एलील का ट्रेट शो ही नहीं होने देगा. तो how do we see या how do we observe के like mixing हो गया? Because colors के अंदर तो हम
yeah. So what we discovered, we found in the progeny, on the basis of phenotype of the progeny, when we took a round, but in the progeny of the round, I have 3:1 round and wrinkled. It means this was impure round. Clear?
But yes, sir. But like my sole question is कि उसकी फिर genotype क्या होगी? How like is it appearing in the phenotype?
इसका genotype, this
जो
one,
improper.
when we say impure round, that means this one. These two,
Yes, sir.
these two, this one and these ones are going to be impures.
So, okay. So exactly, those are impures. मगर उनकी phenotype में we still get the round, right?
How many rounds we will get when this parent will grow? How many rounds we will get and how many wrinkled we will get?
50-50.
No.
1:2:1.
No. Phenotypes. Tell me phenotypes.
If
one parent, this is the same parent, self-crossing. Phenotypes क्या आएंगे इसके?
एक RR होगी, एक RV होगी, एक RV होगी और एक RR होगी small.
Phenotypes क्या बने?
तो 3 round 1 wrinkle.
Very good. तो ये impure ना हो गया?
Yes.
Clear?
But yes, sir.
So we call 1:2:1 is basically genotypic ratio. Genotypic ratio. So underneath 3:1, let me go to the next slide. So underneath 3:1 F2 ratio, basically there is 1:2:1. So this was the phenotypic ratio. And underlying genotypic ratio is 1:2:1. Now, Ibrahim asked question, which I wanted to explain the concept. Now, this factor, they both are controlling what? Seed shape. This versus this, they both are controlling same trait, height. This versus this, yellow versus green, controlling color of seed. This is dominant recessive, dominant recessive, dominant recessive. It means essentially these are same factors but different forms. Today in 21st century, we call them, these are same genes but alternate forms of the same genes. Alternate form of same gene. Only a same gene will be able to contain dictate same phenotype. Is it true? An alternate form of the same gene is called allele. So it means during process of evolution, during this P domestication, the capital R got mutated into small r due to some mutation. And in 21st century, today we know that the round gene is intact in wrinkled plants, but carries a very specific mutation. I highlight mutation by this asterisk. So this gene is silent now. This gene is non-functional in wrinkled. So based on the phenotype, which is wrinkled, it becomes, the phenotype disappears whenever it comes together with round. So we say we represented this form of gene with small r, but only 200 years down the road today, we discovered that, gene is same, but it's a different form of the same gene, which was actually capital R. It's same for these different forms of genes which are controlling different traits. Now, when we have two different alleles existing together, we call this heterozygous state. Heterozygous. What we have? Homozygous dominant, homozygous recessive, and a heterozygous round. If I say heterozygous round, it means capital R and small r alleles are existing together. Okay. Now, what is very important for you and which Mendel did, Mendel started bringing in more predictions. He said, if I take F1 round, cross it with homozygous wrinkled parent, I can predict, he said. And he said, I can predict with 100% accuracy. Can someone of you predict what will be result? F1 round crossed with homozygous wrinkled, what will be phenotype? Just tell me the phenotypes.
Sir, is it 50-50?
This is 50-50. Very good. We call this ratio 1:1, and such a cross where you take F1 heterozygous, F1 was what? Capital R, small r. Homozygous wrinkled is small r. This will contribute what? This can contribute either capital R or small r, and this can contribute either this allele or this allele. When you have this Punnett square, small r, capital R, this, it will be 50-50. In such a cross, when we take F1 heterozygous cross with homozygous recessive parent, such a cross is called test cross. And remember, test cross will always give you 1:1 ratio between the dominant trait, round, and recessive trait. Clear? Is there any question? I would like to quickly go through one last thing to correlate what is this two factors existing together versus knowledge of 21st century. What is happening when I say only half of the factor can be contributed? You should know what is happening here. Is there any question so far?
Yes, sir. I have a question.
Ji, please go ahead.
Yes. I wanted to ask, in the model we are using, we are only taking account two factors. We are calling one capital R the dominant allele and small r the recessive allele.
Yeah.
But in reality, aren't there many more types of recessive alleles which we observe in traits?
So when we say monohybrid cross, monohybrid means we are talking about monogenic traits. Monogenic means only one gene, which is capital R, is controlling the shape of the seed, no other gene. But the capital R in a pea population in different different countries may exist in different alleles. It may have like this, it may have another allele of wrinkled, because whenever you are going to lose function of round, which means mutation in R, and this gene becomes non-functional, in homozygous state, the phenotype is going to be wrinkled. But let's say this is gene dominant wild type or reference, which is the mostly existing in population without any wrinkled phenotype. The wrinkled allele may carry mutation here, one allele. Another allele of the wrinkled may carry mutation here. Another allele, let's say this is existing in Islamabad. This wrinkled is existing in Basel, the city where I did my PhD. This wrinkle is existing in Florida. Okay? Due to mutation, this particular wild type is the gene which performs normal function and which we use as a normal reference, has lost function, and this losing function can be due to a dangerous mutation of this amino acid somewhere else. This could be a nonsense mutation. Okay, this could be this mutation of DNA results in an amino acid change, which makes this wild type protein non-functional. So it means there can be many alleles of the same gene. So R, it can be R prime, R2 prime, R3 prime, R4 prime, as long as they are alleles of the same gene. Did I answer your question?
Yes, sir. Thank you.
Okay. I told you I want to go through one last thing. Is there any other question? There were 3 hands raised.
Sir, okay. I have a question.
Ji, please.
Sir, छोटी सी confusion है मुझे एक, sir, after the F1 generation,
yeah,
when we are getting the other, like in the ratio of 3:1, we are getting the round and wrinkle flowers.
Yeah.
So sir, uske andar you said in the lecture that we are performing the self pollination of them. Sir, लेकिन मुझे इस confusion हो रही है कि मतलब ये क्रॉस नहीं होगी?
नहीं, क्रॉस देखें, क्रॉस एक वो मैंने लेक्चर में पढ़ाया था ना, एक होता है क्रॉस पॉलिनेशन. Cross pollination means I took, the sperm from round plant and I sprinkled it on the stigma of wrinkled. So ये cross pollination होगी. This is a cross between 2 different phenotypic parents. Now, when I take F1 round, which is this individual, and you remember I explained the structure of pea flower in the in my lecture. So in this case, what we are doing, this individual, which is heterozygous round, I'm taking its sperm, which is a pollen grain, and throwing it on its own parent in its own stigma. Now, this is called self-pollination. This is called cross pollination. This one.
Oh, I get it. Sir,
Clear?
मतलब ये कि हम लोग we are just मतलब if we are writing like capital R small r into capital R small r, and we are saying कि self pollination है, तो this process is happening.
Yeah, exactly. वो जो मैंने sign,
I get it, sir.
you remember I had this sign circle with a cross, that is a sign of self-pollination.
Oh, I get it, sir. Thank you, sir.
Clear? Okay, very good
Yes, sir.
that you
Yes, sir.
asked this question. Any other question before I start last 5 minutes and explain what is actually happening?
So different genes and allele are like controlling different types of phenotypes and characters. So is it possible to introduce our own kinds of phenotype? Let's say, for example, if I want to add wings to a human being, or maybe like a firefly, I want to make maybe my pet glow in the dark, maybe. So is it possible?
Yeah, you can. We have plants which glow under UV light. We have uh, mouse who glow green. We have monkeys which we express specific protein. You have to insert specific genes there. Of course, wings is a far-fetched idea in humans, but yes, you can introduce transgene which controls specific phenotypes. Did I answer your question?
Yes, sir. Thank you.
Okay, so now what Mendel discovered, now let's go take a clean slide. When we say, and we said, you know, either this factor or this factor or this one or this one, this is now heterozygous round. Okay.
We have another class which has started.
Yeah, you can watch the recording. It's just last concept. So what actually he discovered, which was not there, and he did not know at all. He did not know all this. It was only discovered in early 1900, I think 1903 or 1904, etc. This was the discovery of meiosis actually. Without knowing, Mendel discovered that what today we know, chromosomes, they exist in pairs. Genes residing on them, and pair of same chromosomes, identical chromosomes are, we learned, we call them homologs. And in this parent, one of the allele is capital R and the other allele is small r. And in DNA replication and cell cycle, you learned about mitosis. In mitosis, a parent gave rise to two daughter cells. If mitosis is happening, it will be RR. Identical chromosomes. Okay. So if we have 46 chromosomes, it will be 46, 46. And this is 2N number, diploid number. We already talked about in mitosis lecture. It's a DNA replication lecture as well. However, when parents contribute to the progeny, the F1, we know the diploid cells that they undergo meiosis and haploids are produced, haploid cells. So one diploid cell, it gives rise to 4 individual cells, each with half the number of chromosomes, and we call them haploids. This was diploid. So what Mendel was discovering or predicted when he said only half of the factor can be contributed in the progeny by each parent, he was discovering unknowingly that chromosomes exist in pairs, and at the time of meiosis, chromosomes are going to go to opposite poles. As a result of meiosis, we will have, you know, capital R going to one pole, the other pole. So this is one end, this is one end, this is one end, this is one end, the haploids. And now this is a random event, whether capital R, and the other parent is capital R, small r, gametes. These are basically gametes. This can fertilize this one, this can fertilize this one, this can fertilize this one, or this can fertilize this one. And the result was capital R, 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 small r, this one. So when we say only half of the factor is going to next progeny, we are actually producing gametes. And when we say we are going to have capital R in a test cross, the gametes for this one is this one or this one. And gametes, because haploids are carrying half of the factor, only one copy of the gene. And these gametes give you then fertilization of these gametes give you then 50-50 ratio. So I would like each one of you to go and look at the difference between mitosis and meiosis and try to understand the homologs and their inheritance during meiosis. And then correlate all this with Mendelian, round versus wrinkle. This is what I want you to learn by yourself. I will share with you link for a short lecture, I think 15 minutes, where I myself have explained what is difference between mitosis and meiosis. But don't restrict yourself to those videos. You can go and watch any other video. So if there is any question, feel free to ask. Okay, so looks like there are no questions. I wish you all a wonderful evening. Allah Hafiz.
Allah Hafiz, sir.